to have some very important guests to come and give a address. Fortunately, our principal is not. To tell you something, why McGill is so well known in research. You see, she's our principal, and yet before this, she was the president of the Natural Science Engineering Research Council that look after all the granting of Canada in this area. And I would like to invite Dr. David Andelman, is Vice Principal, Health Affairs and Dean of the Faculty of Medicine, McGill University since 2012. Before becoming Dean, he served as Director of the Division of Respiratory Disease, Chair of the Department of Medicine, and Physician-in-Chief of the McGill University Health Center.
Merci, Dr. Chang. Bonjour et bienvenue. J'ai l'honneur de vous accueillir ici au nom de McGill et de sa faculté de médecine. Good morning, everybody. I'm delighted to be here today to help celebrate the 60th anniversary of the invention of artificial cells and the contributions of scientists at McGill and from around the world. I'm very pleased to be able to be here together with the esteemed consul and head of Office of Education, Science and Technology and Culture from the Chinese Consulate, Sheng Xiang, Quebec's own chief scientist, Rémi Quirion, distinguished keynote speaker, Professor Zhang and his team members from Peking University, our guests and alumni from here and abroad. I extend very warm greetings on behalf of McGill's Principal and Vice Chancellor, Suzanne Fortier, who, as you just heard, could not join us this morning, even though she was looking forward to being here. But I'm sure she's very pleased to see this excellent turnout. We're very fortunate to have a history of excellence in physiology at McGill. And thanks to strong faculty members and leaders such as Professor Thomas Chang, the amazing person who you just had speaking to you, and the department's chair, John White, who you'll hear from in a few minutes, who've been able to prepare future generations of researchers to help battle some of the most pressing challenges facing humans and their communities. So I referred to Professor Chang as amazing. I think he's sort of a living legend. He's really a trailblazer ahead of his time. In 1956, while still a physiology undergrad here at McGill, his scientific curiosity helped him to invent artificial cells in a makeshift lab in his dorm room. Equipped only with a perfume atomizer, some colloidin, and hemoglobin borrowed from a lab, Professor Chang managed to build a 1 millimeter round plastic cells that contain hemoglobin. He published his first paper in Science in 1964, and in 1989, New Scientist called his student research project an elegantly simple and intellectually ambitious idea that has grown into a dynamic field of biomedical research and development. His original ideas were years ahead of the modern era of nanotechnology, regenerative medicine, stem cells, gene therapy, and blood substitutes. And now 60 years later, Professor Chang has earned his academic stripes many times over. He's currently Emeritus Professor of Physiology, and he's the longtime director of McGill's Artificial Cells and Organs Research Center. He's a member of the Order of Canada, and evidence of his stature in the international scientific community is shown by the fact that he was nominated for the Nobel Prize twice. On a more local level, during our 190th anniversary celebration a few years ago, Professor Chang earned a title amongst the greatest McGillians. And he's been called the Jules Verne of artificial blood research and the red blood cell man. His drive to continue advancing science and improve the health of populations continues strong, as evidenced by this symposium. To quote Professor Chang, "To me as a scientist, what is most important is what is most useful to the patient, not is what is good for your reputation or what pays you the most money. The sick patient should be the most important stimulus for your work." So, Dr. Chang, on behalf of McGill and the populations and communities that we serve, thank you very much for the commitment and dedication to improving healthcare around the world that you've shown. And for inspiring your students, including your first PhD graduate who will address us shortly, Emeritus Professor and Director Mark Poznański from the Robarts Institute, who will really show how Professor Chang has allowed greatness to go from generation to generation. We look forward very much to hearing your anniversary lecture, Professor Chang. Alors, je vous souhaite à tous un symposium intéressant et enrichissant. Merci beaucoup.
Thank you, Dean Alderman. Thank you for all the kind word. So may I introduce now Professor John White. He's the new chairman of the Department of Physiology. He received his PhD from Harvard in 1986. He has been a member of the Physio Department at McGill since 1991. Since coming to McGill, he has made important contribution to our understanding of the role of vitamin D in human physiology. His research opened the field of study of vitamin D as an inducer of human antimicrobial immunity and has provided fundamental insight into the mechanism of its action as an agent of cancer prevention. Professor White.
Well, thank you, Tom, for the opportunity to speak here today. As Tom mentioned, I am a recent chair of the department. It's been a big year. I turned 60 this year, just like artificial cells. And I have to say that artificial cells are looking much fresher than I am. As Dean Eidelman mentioned, the Artificial Cells Project started as an undergraduate project in the physiology department. If I understand correctly, Tom was one of the guinea pigs in that respect. He was one of the first 4 students who did an undergraduate honors project in the department. And that was an initiative of Hank McIntosh, who was chair at the time and probably one of the more dynamic chairs in the history of the department. And after he got his MD, Tom turned artificial cells into a PhD project. The problem was, who's going to supervise it? The project was so avant-garde that really the only expert in the world was Tom himself. I gather that Hank McIntosh agreed to supervise the project, perhaps with some reluctance because it wasn't exactly his area of expertise, but obviously the project worked out extremely well. And as you are all aware, it turned into a single author paper in Science published in 1964 that has now been cited 1,200 times. The rest, as they say, is history. Tom became a faculty member in the Department of Physiology in 1966 and rapidly became a full professor. And in 1979, the Center for Artificial Cells and Organs in the department was started. And since then, of course, as everyone in this room knows, it has become an international network. The journal that grew out of it, Artificial Cells, Nanomedicine, and Biotechnology, has been ranked by Reuters as 4th out of 77 journals in the field of biomedical engineering. I am very proud to be chair of the physiology department, and I'm even more proud that Tom Chang has been a pillar of that department since 1966. I really think that this symposium is a fitting honor to his scientific legacy, and I wish all of you a stimulating and thought-provoking time. Thank you very much.
So now I want to introduce Max Poznański. Mark Poznański worked as a project undergraduate student with me, then became my first PhD graduate student. He then spent his postdoc at the laboratory of the head of biophysics at Harvard. He wanted to come back to McGill, but at that time, there was no position in this area for him, so he became associate professor at the University of Alberta at Edmonton and quickly became full professor, then vice dean of research. He was so good that he was recruited by the well-known Robert Research Institute of the University of Western Ontario as their director and full-time professor. He held this position for many years until his retirement recently to become president of his own consulting firm. He is also a member of the Order of Canada. Mark?
Professor Chang, guests, it's an honor to have been asked to represent the hundreds of graduate students, postdocs, and trainees of the Chang Laboratory over the past 53 years. In 1963, as a young undergraduate student in physiology at McGill, I had a dilemma. With little knowledge, I had to choose an undergraduate supervisor, and the pickings were significant, as it was a large, active department with 3 fellows of the Royal Society of Britain, F.C. McIntosh, B.D. Burns, and A.S. Burgeon, and a host of well-trained and well-funded young investigators. And then there was Tom Chang, a newly minted MD PhD working on artificial cells. He wanted to create completely artificial entities to replace biological cells that were either absent or not functioning. He wanted to create artificial blood to be used in place of transfusions, especially in areas where fresh blood was difficult to obtain. He wanted to produce cells that could help detoxify blood in patients in kidney failure. Now, realize for a moment that in 1963, the structure of DNA had just been announced, and there was no such thing as genetic engineering. The first stem cell papers had been published, but really only as a concept. High-risk organ transplantation had started a few years earlier, and regenerative medicine was not yet on anybody's lips. But this young McGill faculty member's enthusiasm was contagious. He had a vision, and he had a pretty good start. He succeeded in creating semi-permeable aqueous microcapsules, which in essence could function as artificial cells to mimic red blood cells if they were filled with hemoglobin, or filled with a particular enzyme for the potential treatment of enzyme deficiency diseases or inborn errors of metabolism, the project that Professor Chang assigned to me as an undergraduate. Now, if the truth be told, at least in Canada, Tom's ideas were met with a huge amount of skepticism. But he had, and this is very important, he had a few senior people who believed in him and gave him a break. There was Hank McIntosh, a huge force in Canadian and British science in the 60s, who was chairman of the department at the time and had been Tom's thesis advisor. And there was Sir Malcolm Brown, president of the Medical Research Council of Canada. Today, we understand the extent to which Tom was at the cutting edge of developments in biotechnology and nanotechnology, except for the fact that those terms or disciplines had not yet been defined. Skip forward a half century, Professor Chang is still in his lab at McGill working on the use of artificial cells to solve particular problems in medicine. But now the world's caught up to his revolutionary ideas, and the entire area of nanotechnology represents one of the most exciting technologies of the early 21st century, not only in medicine, but in any number of other industries. By 2010, the entire genome had been well defined, and the genomes of many different organisms had been sequenced. And many technologies associated with genetic engineering, including gene editing, had been demonstrated. And the entire area of artificial cells, championed by Professor Chang in the 60s, was no longer a dream or some sort of futuristic objective. In 2011, Craig Venter, one of the major players in the Human Genome Project, announced that he had created or manufactured a completely synthetic line of live cells. The DNA code came from his computer. A long strain of DNA containing the necessary genes was synthesized and placed into the empty shell of a bacterium, from whence it proceeded to both replicate itself and create the proteins necessary to sustain life. With these cells, Venter anticipates being able to produce cell lines that create vaccines or specific medicines or anti-cancer therapies or even nitrogen-fixating bacteria that can be used for the replacement of expensive and sometimes inefficient fertilizers. With the use of modern genomic and computer technologies, Craig Venter had succeeded in creating the same artificial cells with the same objectives that a young Tom Chang at McGill had proposed and developed, albeit with much less sophisticated technologies, some 50 years earlier. In every sense, Tom is a father of not only artificial blood substitutes, but really the entire area of nanotechnology. Finally, from a personal comment about Tom Chang, as a teacher and as a mentor. Even as a newly minted professor, he had a sophisticated Socratic teaching method. I'm not sure he was a student of Socrates, but at least he used the technology. Late in the afternoon, we would be discussing a research problem, and he would challenge me. He would say, I can think of 4 or 5 ways of going about solving the problem. And without divulging any of them, he would say, let's discuss them in the morning. I would spend the evening thinking about them, about his 4 or 5 solutions, and in the morning we sat down to discuss, and maybe I would have figured out 2 or maybe 3, and if I was lucky, and on occasion I even thought up of an original one. This was what it was like working under Tom's tutelage, and on behalf of all his trainees, I thank him. Thanks, Tom.
Thank you, Mark, for all the talk about your experience. Okay, the next presenter, it's a pleasure to introduce Professor Remy Quéland. He is the first chief scientist of Quebec, which means that he looks after all granting agencies in the province of Quebec. At McGill, he is a full professor in psychiatry. He was vice dean, Faculty of Medicine, and a senior university advisor in health science research. He was also the scientific director of the Douglas Mental Health University Institute at McGill, and also the inaugural scientific director of the Institute of Neurosciences, Mental Health, and Addiction. He received many awards, including Chevalier of the Ordre National de Québec and a member of the Order of Canada. Professor Quéland.
Merci beaucoup, Tom. C'est un plaisir au nom du gouvernement du Québec d'être avec vous ce matin. So, as was mentioned by the colleague, Dr. Chang is a real pioneer, like you will not meet so many times in an academic career. For me, I was at McGill for close to 30 years. Tom Chan was at the comparable to a household name like Brenda Milner. For those of you that know neuroscience a little bit, Brenda Milner, pioneer in learning and memory. She will turn 100 in a few months. She's still active. Charles Taylor, a philosopher at McGill University, and Tom Chan, a superb trio with international reputation and leadership for almost 65 years. Today, as a chief scientist of Quebec, as was mentioned, I oversee the activity of our 3 funding council in health, in science and engineering, in social science and humanities. I think that Mark already mentioned how far ahead Tom was. One of the things that we're trying to do at the Quebec funding agencies is to make sure that for a huge challenge of our society, like building artificial cell, we have experts from many disciplines working together. So almost 60 years ago, Tom was already doing that. But today, it's still a challenge for many scientists in academia to work together on very significant problems. Just think of climate change, just think of aging. Yes, you need healthcare expert, but you also need engineer, you need chemists, and you need social scientists. So one of the recent programs that we developed at the funding agencies is called AUDACE. So to have audacious, very far-thinking program, having experts from different disciplines applying for grant. And if I use a baseball analogy, not to get to first base, but to have a home run. So I think that Dr. Chan should apply for one more grant from us in this type of program, and I'm sure that he'll be very successful. Merci beaucoup et bonne journée.
Thank you, Professor Quirrell. It's a pleasure to introduce is the Council and Head Office of Education, Science and Technology and Culture of the Consulate General of China in Montreal. 2004, for 4 years, he was Council of Education Office, the Consulate General of China in Chicago, USA. 2005, for 5 years, he was the Director of the Chinese Service Center for Scholarly Exchange, Ministry of Education, and since 2014, he holds his present position. Dr. Xiu, please.
Distinguished guests, ladies and gentlemen, good morning. On behalf of the Consulate General of People's Republic of China in Montreal, I would like to take this opportunity to extend my heartfelt congratulations to Professor Chong. I would like to also take this opportunity to extend my cordial welcome to all the delegates, including overseas Chinese and Chinese delegates. 2 years ago, when I came to Montreal to work at the Consul General, I worked as Counsel for Education. My predecessor, Mr. Zhai, told me there is a very famous professor in McGill. You should meet him sometime. When I first met Professor Chong, I'm deeply impressed by him. I'm deeply impressed by his spirit in research. So when I go to McGill, every time I tell the Chinese students in McGill, today you are proud for McGill. Tomorrow, McGill will be proud for you. You should learn from Professor Chong. So I'm very proud Professor Chong made so outstanding achievements. I would like to take this opportunity to extend my heartfelt congratulations to Professor Chong. Congratulations.今天呢，在座的有很多来自中国的专家学者，那我很高兴的借此机会代表中国驻蒙特利尔总领馆，向大家呢表示热烈的欢迎，向张教授表示衷心的祝贺。我们为有这么一位优秀的华人专家学者感到呢非常的自豪。我希望所有的中国学者能够像张教授一样取得更好的成就。So, because artificial cells is so new to me, I have nothing to say except my cordial thanks. My congratulations to Professor Chong. I hope every delegate make great achievements like Professor Chong. Thank you so much.
Thank you very much. Thank you. I'm going to tell a story. It's not a scientific lecture. Later on, I'll be giving scientific lecture. So my present position at McGill and also outside McGill. And, okay, so as Dr. White mentioned, I was one of the 4 guinea pigs in McGill's new program of honors physiology. You notice where they put me? In the weakest chamber of the whole heart. They put Hugo in the biggest heart, left ventricle, and Morty in the other strong heart. Look at me. But what they don't know is that I control the sinus auricular node with all So there are excellent people in physiology. So one of the persons that really stimulated me is Professor Arnold Burgeon. He gave the advanced course in cell physiology, membrane transport mechanism, and his lectures so stimulated me that I thought, well, with all these basic science, why don't we do something called artificial cell? And it's based simply on an ultra-thin synthetic membrane with intracellular content that doesn't come out, but what's outside also cannot get in. By adjusting the membrane permeability, you can have things you want to go in, and things to come out. I have made the mistake of asking different people and telling them what I plan to do, and that was the reaction. And they thought it was very funny. They are very polite, though they don't say I'm crazy. They just say it's funny. So to make things, you see, I come from a family, my grandfather started as a preacher, son, very poor, and he struggled, started everything himself to become one of the richest men in the city. So he told me, don't worry what people tell you. You can start anything you want, just do it. So I went to Douglas Hall, resident, where I stay, and there I was able to use my dormitory room to do the research. Now I have to be very honest with you. That's not me. He looks much more handsome and younger than me. This is a BBC from London coming to take a video, and he looked at me and said, "You're too old, you're not handsome enough. Get me somebody." So I got my son. It's a bit of a prejudice. My son is in the space agency, so he's advanced, So he took over the thing, okay? So anyway, that's the type of facility I have at that time. Now, so I was messing around and got some preliminary data, and in physiology, honest physiology, you need people to do research, and they assign you a project. But like my grandfather teach me, don't be shy, go and ask people what you want to do. So I went to Professor Burgeon. I wrote him that I'm going to have a meeting, and then he doesn't want to trouble now, he's a bit older than me. So I asked him to send me his photo. So he not only sent me the photo, he also make a statement. He said that, I still recall you coming to see me and saying that you prefer to do a different project and would like to try and make artificial blood cell. I think I said, go ahead without much expectation, and et cetera. So it was very nice of him to say that. And so these are the things I did in the physiology department when I was in the project. And I was able to make hemoglobin artificial cell that carry oxygen with the oxygen dissociation that's not too much unlike. And the test was the department asked me to give them a sample, and they asked Professor Seckley, an expert in oxygen transport, and to repeat this thing. I had a few nights of no sleep, and finally they told me, yeah, he got more or less the same thing, you passed. So now, then I start medical school. Hank McIntosh let me continue, and the Faculty of Medicine give me a summer fellowship, so I was able to continue. But I also meet a very strong supporter of this idea, and this is somebody called Nancy Yan. And she's from Ottawa Civic. She got a scholarship to come to McGill, and she made the biggest mistake of her life. She became Mrs. Thomas Chair. I was so busy combining research and medical school, and then afterwards too, that we postponed our honeymoon. I'll tell you when we have it. And then she was a general nurse, despite offers of administrative position, so she had more time to mow the lawn, shovel the snow, and do the housework. So she said, I can have all my energy on my research. So whatever I say and my research result, half of it goes to her. After making medical school, I went to see Professor McIntosh to see whether I can take a PhD in physiology. He looked at me and he said, well, I don't know. This is not the type of research for PhD. It's too different, but it's a good project. But who are going to find to do the supervisor? Nobody here does it. I said, of course, you can do it. Yeah, you are the chairman, you should take the responsibility. He said, I'm a neurophysiology, But Hank is very nice and he's very forward-looking, so he agreed to do that. In fact, okay. So after working, I got the artificial cell that has very thin membrane. If you break it, it's nylon and protein, so it's very strong, very thin membrane behind. You put it in salt solution, it shrinks. You put it in hypertonic solution, it returns the shape. So after PhD, I went to Hank McIntosh, and I said, okay, I published a paper in Science, and lots of people in the state offered me a good position. What about position in physiology? I don't want to run around. I figure I want to continue the research here. And he said, well, look, I helped you so much all along. This is too difficult for me because there's no money for such a position, but I can do what I can to help you. Then come Professor David Bates. He elected to become a physiology chair here. At that time, he was Associate Dean of Research in Medicine. So he said, Tom, why don't we try the Medical Research Council? They have a junior career investigator that pay the salary, and you don't have to worry about the salary. So for 3 years, I got the MRC scholar, and then as MRC Associated Career Investigator until they moved me to the new McIntyre building they just built. And Professor Bates put me in the biggest lab in the whole building. And later he expanded more, so I wasn't too popular with the physiology department staff. Now, the thing is that, you see, I have very good facility. It's a bit better much better than the facility I have in the dormitory room compared to before. Now, this appointment is excellent because it allowed me to do anything I want as long as they renew it after 5 years interval. And what's good about this, I don't have to worry about the research I do. Our earlier attempt was to reproduce biological cell, for instance, complete red blood cell or artificial liver cell, and also membrane transport mechanism. So I decided I should not be preparing biological cell or replicate of biological cell because culture or God or cell culture can do a much better job. So why try to fool around? So instead of doing this, if that's the case, why do you want to prepare artificial cell? Well, the thing is that you can go outside the box and innovate with unlimited variation in content, size, and configuration. For instance, like the beginning, you can put cell, hemoglobin enzyme, magnetic material, adsorbent, and other things inside the artificial cell. To prevent them from coming out, things from outside can be excluded, but molecule can go through. And jump ahead to now, with the research from our center, from all around the world, this is what happened. You can put almost anything inside this artificial cell. I don't have time to go over everything. You can also change the dimension from macro, bigger than cell, micron, nano, and soluble nanobiological polymer. You can vary the membrane to different changes. Now, because of the different changes and configuration, not limited by preparing biological cell, you have unlimited use, only limited by imagination and grant support available. For instance, you can do different things. I don't have time to go over each of these. Maybe we take 2 examples and look at this. Let's look at this one. I always choose the simplest one for development towards clinical use because the simpler it is, the better chance and the less time you have for clinical use. So let's look at this. That's an artificial kidney. A naive researcher always can think of naive idea. I thought, why don't we make something this small to replace the whole artificial kidney? To do this, let's look at the theory. I use a slide rule. I don't know whether any of you old enough to remember slide rule. And I did some calculation, and I found that 10 cc of artificial cell has 100 times more transport rate than a whole artificial kidney because of the dimension and because of the ultra-thin membrane. This being the case, then, the things can move across very quickly as a cell. But then it's such a small volume, you can remove too much, so you can put in different things. And it will become a microdialyzer bioreactor combined. So look at adsorbent, the simplest one. The thing is that if we use adsorbent, adsorbent can release powder, it can remove a blood cell. So what I did is that I put an ultra-thin membrane around it, very thin, 10 nano dimension. It doesn't, powder doesn't come out anymore, the blood cell doesn't go in anymore, and the permeance toxin can go in. That's the theory. You can make this into a very small system and put it in the column. There are millions of this, but this is schematic. So things from the patient can go through, cleanse, and then go back to the patient. Now, I present this paper in the Artificial Internal Organ Society, and Professor Koff, who is the inventor of artificial, I should at this point say that the reason I can do this research is because I'm lucky. I happen to meet people who are forward-looking and willing to help, and these, as Professor White said, are very senior people and very helpful. So you would think he's the inventor of artificial kidney, so he would not like something to replace artificial kidney. But no, he's very forward-looking, and he heard my lecture, and what he did is he wanted me to join his group, but I didn't want to, so he wrote a strong letter to the Medical Research Council and also McGill, saying that if you guys don't support Dr. Chang with big grant and big research, I'm going to get him to come to my group and start the clinical trial here. So the result is I got a very big special grant. Malcolm Brown, as just mentioned, the president of MRC, started a new special project and allowed to do basic fundamental innovative research. Now, the research is as follows. The thing is that if we use adsorbent, adsorbent can release powder, it can remove a blood cell. So what I did is that I put an ultra-thin membrane around it, very thin, 10 nano dimension. It doesn't, powder doesn't come out anymore, the blood cell doesn't go in anymore, and the permeance toxin can go in. That's the theory. So in practice, I found that when you do this, if you don't cover it, particle get released. If you cover it, the particle doesn't come out. So I started the hemoperfusion symposium very much like the blood substitute symposium around the world every year, 2 years, to stimulate research in different parts of the world. And everybody found that by coating, yes, it prevents release of powder, but the membrane being polymer is not blood compatible. In many cases, it traps cells, especially platelet, and many people thought that after long time research, they thought that maybe we should drop the idea. It's a bit like the blood substitute thing because of all the problem. But I always find that problem is a challenge, and challenge is something you can use to move forward. So after a lot of research in the lab, I found that, I keep saying I because at that time I was the only one doing research. That was before the big one come in, where we have a very big group. Now, I put human albumin that get adsorbed to the surface, and then by doing this, the cell going through the outside don't recognize the membrane, they only see human albumin. So as a result, as you can see here on the right side, there's no trapping of red blood cell or platelet. It's very clean, and this is a normal red blood cell sitting on top. So if we do this in patients, it was found that there's no significant decrease in platelet. Without the albumin, it will go down to about 20%. So now we want to test and see whether artificial cell in this form can compete with the artificial kidney. So the simplest thing is always good to start with simple things, and where you can do things quantitatively. And furthermore, I have a own idea that I thought it's better to prefer clinical trial design to show superiority and not equivalency. You can more conclusively demonstrate. So over the next 3 years, I was on call 24 hours a day because clinicians, they don't want to be responsible for liability because I make this in the lab and bring it to the hospital. They figure I should be responsible, so I get to do all the clinical trial directly, and they do the clinical analysis. So now, this is one typical example. A patient comes in with 3 times lethal dose of sleeping pill. After dialysis for 5 hours, she remained comatose, hypotensive, and cardiac arrest. They have to restart the heart. So this is one of the cases. They phoned me right at the beginning so I can get everything ready and wait to get. So when I put this on the patient, the level come down and everything become normal. I should say that these are clinical part is analyzed by the people up there. They're on nephrology, so they do independent assessment of the clinical result. You notice Paul Berry, he was one of the early residents, was most enthusiastic. That's why I put his name there. He's now the chief of dialysis at McGill. So the hemoperfusion then show superiority over hemodialysis in all the treated patients. So as a result, it's approved for routine use by Canadian agency and US FDA. So I told you about honeymoon. Now we have time to go for honeymoon. It's a long time after we got married. So now, then one of my oldest sons become what you call a computer nerd. So I said, okay, Harvey, Harvey is now an assistant professor in medicine at the Jewish General Hospital. So he put in the data into the system and said, there, you make a mistake. It's not 100 times. It's only for 90, the big microcoefficient. If you use a small one, like what you have, it should be 200 times. So I feel good. The slide rule is not that bad after all. So, and after that, you see, as Dean Eidelman said, I'm not for profit, I'm not for, so I go around the world to show how to do things. And I, for instance, went to China to give lecture, and Professor Beng, a very well-known scientist, and then give lecture to show how to do it in hospital, and also go to different other places in Israeli Institute of Technology and other places all around the world. So these also, here's on the left side is Professor inventor of artificial kidney, so they even get me to get involved with their society. Now, then come Dr. Sam Freeman. I emailed him and got him to send me his photo and invite him to come, but he feels that his mobility is not as good, so he doesn't come, but he sent me a nice photo, and when he was Dean of Medicine, he recognized this as the Artificial Cell and Organ Research Center. And then when he becomes vice principal of McGill, he supported my application for the Viral Center of Excellence in High Technology. I told Professor Quiron about this one. It's a very, very difficult, competitive thing for all centers. And fortunately, we got a place here. And 5 years, the criteria is for me to train 20 PhD students in 5 years. So it's a little bit of hard work, but we did it. And they like the result, so they have this as full permanent position in professional assessment in the university budget. And these are the people that work in the lab around that time. You recognize them, they have a red name in their deck. So going further, Thurman, remember we put albumin on the surface, and Thurman from the US found that this column can remove selectively albumin antibody. Going further, other people, including the late Professor Yu in China and many, many others, can use other antibody, other antigen, and use it for lupus and other things in clinical trial. So other uses for hemoperfusion include uremia, intoxication, hepatic failure. And in 2017, usually when we finish the clinical trial, I move away to another area to do clinical research and let the people do the more innovative to expand the area. So these are all senior scientists around the world, in China, in Russia, in Japan. So all their work since that time result in tremendous progress, and now you can have this in different area of application. So So this is looking at biotechnology and nanobiotechnology, nanomedicine. Of course, we don't have time, so we just mention one thing. As I said, we only mention two things in this discussion: blood substitute and oxygen therapeutic. The opinion during 1964 was that there's no need for artificial red blood cells because donor blood was good enough. Yes, it was, and I should share the same blame because that's what I thought too. That's why I thought we should go and look at other things. Attention tuned to the other uses of artificial cell. Now, when 1987 HIV came, there was nothing to replace the infected donor blood. So urgent world effort only started much later for something to replace HIV. And at that time, around the world, many of you are sitting here, started to work on this because of the urgency of people getting infection. So these are all the most of the people at that time, you recognize your name here, that's doing this research. And these are some of the more senior people. These are the keynote speakers at the International Symposium Blood Substitutes in Beijing. You notice many of them are here, and you are here. And this is the meeting we held in Harvard. He was the chair, and these are many of the people at the meeting. So in summary then, blood substitute, after all this research, we found that it's like any other type of medical therapy. You need lots of fundamental applied and clinical trial before ready for patient. You can't do it overnight. So let's look at one example. Hemoglobin molecule can be a sample and crossing together by biphonal engine. One of the popular ones is glutathione to form polyhemoglobin. That was way back in 1971. Now, because of the HIV crisis, many people then put in a lot of work, and BioPure and Northview in the US independently on their own. I'm not their consultant. They are completely on their own. Develop polyhemoglobin and use pasteurization to inactivate HIV crisis. And jumping forward, after lots of work, 2002 approved in South Africa for some surgical uses to replace HIV-contaminated blood. Thus, the initial aim of replacing HIV donor blood was a 10, but it's 15 years too late for many HIV-infected patients. So HIV in development countries no longer a major problem because we have good screening tests. So now we're looking into other important uses. One of this is stroke and heart attack. Coronary artery and supply to the brain could be blocked by arteriosclerotic plaques and other things, so preventing normal passage of red blood cell and thus ischemia. Theoretically, everybody, including me, thought that if you then perfuse hemoglobin solution, polyhemoglobin, that's a solution, it has no problem going through. It does, but the sudden influx causes oxygen radical ischemia reperfusion injury. And this is because of the tissue injury, removal of nitric oxide, resulting in all the problems. Of course, there are other reasons, but these are some of the reasons. To solve this problem, Daniele Felici, doing his PhD, we tried to incorporate the antioxidant enzyme into here. And by doing this, there's no problem related to this. I have to say here that all my good students are grabbed by other lab. They don't let them come back. They all stay there. So he's now in NIH, FDA, very high senior position. So Pawenta did his master, showed that in fact this can prevent edema in the brain by preventing ischemia reperfusion injury. This is polyhemoglobin. This is when you have the enzyme. We also explored the potential use of this approach for organ preservation, for transplant. In this case, intestine, which is a very sensitive organ. It is shown that with the use of the antioxidant enzyme polyhemoglobin, oxygen radical is prevented from forming compared to polyhemoglobin alone. Now, let's look at severe blood loss. Now, transfusion in emergency, you have to wait for the ambulance to come and then move the patient to the hospital, have it cross-match, and depends on the weather condition. If you're in a war zone, that might take a long time. And finally, you get transfusion with a lot of delay. Now, polyhemoglobin has no blood group, can be given right on the spot. So being the case, then you can delete all these and do this right away. Furthermore, you can also put medics with a backpack with this type of solution in the back, use helicopter, put them down, they can run around and transfuse patients. So, is this possible? Well, in more and with the GHOST group, ambulance patient, where they give the transfusion right on the spot, randomized into two groups. Of course, I'm oversimplifying this. One group is control, saline, normal treatment. The other group is polyhemoglobin. In this group, they have to give blood transfusion on reaching hospital. This group, no need for blood transfusion for 12 hours. However, this group, 0.6% non-fatal heart attack. This group, 0.3% non-fatal heart attack. So FDA did not approve this for routine use. So because of this, many groups around the world, including many of you sitting here, start to analyze how to prevent this problem. There are different approaches to solve this problem. In our case, we looked at publication from Sweden, and they found that increased tissue PCO2 is equal to decreased survival in hemorrhagic shock. In Denmark, they found that increase in PCO2 is related to myocardial ischemia. So looking at all these, Bian from the Tsinghua University from China, very well-known bioengineering school, when she came and said, look, let's put the enzyme carbonate hydrase to the other two enzymes to remove the carbonate, the CO2, and see what happened. So this. The aim, again, is to show superiority. So instead of giving the same enzyme concentration as blood, we prepare these with 6 times the concentration of red blood cell. Enzyme is only a small fraction of the content. So by increasing 6 times, you're not changing too much. So let's look at, this is the most important question, is the heart. So we look at the recovery of heart ischemia, and this is the result. This one, saline, not much effect. Polyhemoglobin, some change. And blood, yes. But this one is superior to blood, superior to polyhemoglobin. You notice comparing blood to polyhemoglobin, blood is better. So that may explain, could be one of the reasons why there may be side effect, because your patient that come in are in different conditions, different. We also look at the intestine. We purposely use 90 minutes hemorrhagic shock to go beyond the 60 minutes safety window. I always believe in doing the severest test. So when you do this, I don't want to go into the detail, you find that this one has no problem. The blood has only some effect on the epithelial cell, and the polyhemoglobin has a bit more program. So in other words, it seems that would help also in the intestine, which is one of the problem of irreversible shock in long-term sustained ischemia. So summary, laboratory results look promising, possible to show superiority over red blood cell in sustained severe hemorrhagic shock. I should emphasize severe hemorrhagic shock and sustained, because in less severe case, you do not need the carbon dioxide removal system. The red blood cell, there's enough red blood cell to do it. However, translation to clinical, you need a lot more production, preclinical testing, detailed clinical trial. So this look promising, but a lot of work needs to do. We shouldn't wait again until it's too late. So you can look at the result. Everything I publish are put online, so you can look at this. We have discussed in detail the use of artificial cell for micro device and nano device in the form of hemoperfusion. This is an example of extensive support for basic fundamental research before use to a patient for clinical trial and clinical use, resulting in rapid approval. The second example of blood substitute is the example where there was not enough fundamental basic research until urgent need for blood substitute because of HIV. As a result, the basic research support was skipped and went directly into production for clinical use, resulting in many problems. Let us now go very quickly and very simply into a few other areas, very briefly. For instance, enzyme and gene therapy based on biotechnology and nanotechnology. Inside artificial cell, enzymes are prevented from immunological reaction but can act on substrate diffusing in with product diffusing out. We have explored a number of potential uses, including urea removal, inborn area of metabolism, lymphosarcoma, melanoma, bilirubin removal, multi-enzyme system with cofactor recycling, including urea to essential amino acid conversion. Yeah. So let's jump quickly to this one. So this article was published back in 1976. We can use biodegradable microcapsule particle to have enzyme, hormone, vaccine, and other biologic. And the biodegradable is that they can be converted to lactic acid and finally into water and CO2, so nothing gets accumulate in the body. And this is a release of insulin, slow, very slow, and very, very slow. And nowadays, you can use this for nanocapsule, polyaerosol, and nanoparticle, and you can also change the surface for target delivery. And something exciting also is done by Jan's group that has nothing to do with artificial cell, but he used the biodegradable polymer with slow release mechanism to connect a small nerve on the normal side to the paralyzed nerve on the paralyzed side. And you'll hear his lecture later. And way back here, I also found that you can put magnetic material inside artificial cell with biologic, so you can use this for different things. I don't have time to go over the detail. But one example is that in tumor cells, you can inject nanoparticle with magnetic thing, and also radioisotope, and also chemotherapeutic drug. You can accumulate the nanoparticle by putting a magnet over the tumors, and many people are doing this around the world. Okay, now, very quickly, just jump over this one. So nanomedicine now is a very large area, and again, this is free for all to look at. Cell and stem cell therapy. Way back, 64, I found that it's possible to put cells inside artificial cells. Okay, so this is one of the things that I did, that cells is excluded from immunological reaction. And different things can be put in, islets, hepatocyte. I went to Conard and get them interested in putting islets because they are the expert, as I said, are always expert, do the research, and they do some very good work and stimulate ongoing research by many groups solving problems. Genetic engineering cell is another area. Concerning cell is now a worldwide study. There are cells, tissue, and there's genetic engineering cell, microorganisms. Artificial cell containing microorganisms include removal of cholesterol, conversion to L-lysine and L-dopa, a genetic engineering cell for removal of urea in rat study, lactobacillus from yogurt modified to be able to remove urea. Because of the safety of lactobacillus, this was extended by Prakash to bile acid deconjugation to remove cholesterol. And stem cell. Let's look at stem cell because he's going to talk. Stem cell, Dr. Liu from China, when he was here, research associate, we put stem cell inside artificial cell, exclude it from immunological problem, and it can release hepatic growth factor to, in the peritoneal cavity, it drains directly into and stimulate the recovery to full function. So this is not my field, but lots of people are doing this, and some people from Russia told me that they are using artificial cell to do caviar. I don't eat caviar because so other people are using for aquatic culture to future and microcomputer and nano robotic. That's not my area, but a lot of people are doing this. So in summary then, objective is not to prepare biological cell. God, nature, and cell culture can do this better. Artificial cell is to go outside the box, to innovate, no limit in variation, content, dimension, configuration. As a result, applications are only limited by one's imagination, ability of research support. And these, I should say that I'm giving this lecture on behalf of many of the people that do research in the lab. I already showed this beginning slowly, but this we can go faster. Okay, thank you very much.